Hello friends, Stephen Moulton Babcock was American agricultural chemist. He is best known for the single grain experiment. This experiment tested if the cows could survive on single type of grain through the feed. It would lead to the development of modern nutritional science for the cattle. It was carried out at the University of Wisconsin-Madison from the year 1907 to the year 1911. The most significant name for the experiment is Stephen Moulton Babcock. The day of 22nd October is the birth anniversary of Stephen Moulton Babcock. It is an excellent opportunity to discuss the contribution of Stephen Babcock to the science. In the year 1881, agricultural chemist Stephen Moulton Babcock returned to the United States after earning his doctorate degree in the field of organic chemistry at the University of Göttingen, Germany, to accept the position at the New York State Agricultural Experimental Station. His very first assignment was to determine the proper feed ratio of the carbohydrate, fat and the protein from the cow feces using chemical analysis. His finding determined that the feces chemical composition was similar to that of the feed, with only the major exception being the ash. His results were found similar to the German studies done earlier. This led Babcock to think about what would happen if the cows were fed with a single type of grain of cereal crops like barley, corn, wheat. Stephen Babcock then, 7 years later, he accepted the position at the University of Wisconsin-Madison Agricultural Experimental Station as the chair of agricultural chemistry department. Immediately, he requested to the William Arnon Henry, Dean of Agriculture and then station director. But Henry refused. Babcock continued pressing Henry to perform the single grain experiment. He also requested to J.A. Craig, head Department of Animal Husbandry. He also refused the idea of single grain experiment. In the year 1897, Craig was replaced by Carlyle. Carlyle was more receptive to the Babcock's idea, initially trying a salt experiment with 8 dairy cows as a matter of test performance, while the 8 other cows received no salt. One of the cows from the group not received the salt was found dead. Carlyle discontinued the experiment and all the remaining cows were given salt in order to restore their health. In the year 1901, Henry chaired as the Dean of Agriculture. He approved the permission to perform the experiment. Carlyle approved the experiment with only 2 cows. One cow was fed corn, while the another was fed rolled oats and the straw, with the hopes that the experiment would last for 1 year. 3 months into the experiment, the oat-fed cow died, and Carlyle halted the event to save the cow's life. The results were not published mainly because of no data was listed by the Babcock. In the year 1906, a chemist from University of Michigan, Edwin B. Hart, was hired by Babcock. Hart previously had worked at the New York State Agricultural Experimental Station and had studied physiological chemistry. Babcock and Hart planned for the experiment. The single grain experiment was thus born in the year 1907. From the year 1907 to the year 1911, the experiment was carried out with Hart as a director, Babcock providing ideas, and Humphrey overseeing the welfare of the cows during the experiment. Chemist Elmer Malcolm helped for the analysis of the grain ration and cow's feces. The experiment called for 4 groups of 4 heifers, each during which 3 groups were raised and 2 pregnancies were carried through the experiment. The first group was supplied with corn in the form of stover, cornmeal and gluten. The second group was supplied with nutrients from the feed in the form of straw, wheat meal and wheat gluten. The third group was supplied with the nutrients from the oat plant in the form of straw and the rolled oats. The last group was supplied with the mixture of the other three. The experiment was carried out with Hart as director, Babcock providing ideas, and Humphrey overseeing the welfare of the cows during the experiment. Malcolm was assigned to analyze cows' feed and animals' milk, blood, feces, and the urine. In the year 1908, it was shown that the corn-fed animals were found the most healthy of the group, while the feed-fed groups were the least healthy. All the four groups bred during that year, with the corn-fed cows being the healthiest, while feed and mixed-fed calves were stillborn or later died. Similar results were found in the year 1909. In the year 1910, the corn-fed cows had their diet switched to feed, and non-corn-fed cows were fed with corn. This produced unhealthy cows for the formerly corn-fed cows, while the remaining cows produced healthy calves. In the year 1909, feeding patterns were reintroduced to the respective cows. In the year 1911, the gestation results of the 1909 reoccurred. The results were published in the year 1911. This experiment made it clear that there were marked differences in the nutritive values of which not detected by any chemical means available at the time, and that current scientific basis of the formulating rations were seriously inadequate. More important, the experiment led to the conviction that simplified diet must be used for the solution of nutrition problems. The experiments would lead to the development of nutrition as a science. It also lead to the determination that there were minerals and the vitamins in the food. That research would continue into the year 1930 and the year 1940, ending in the year 1948 with the discovery of vitamin B12. Determination of butterfat content of the milk is also concerned with Stephen Moulton Babcock. He developed the famous Babcock test, which determines the content of buttermilk in the milk, butterfat in the milk. He then worked with the bacteriologist Harry Russell in the development of process of cold curing for ripening of the cheese. The Babcock test set worldwide standard for butterfat determination of the milk, while the cold curing process enabled Wisconsin to become the leading cheese producer in the United States. Before the Babcock test, dishonest farmers could water down their milk or remove some cream before selling it to the factories because milk was paid for the volume. Honest farmers, on the other hand, as well as those that produced a naturally rich milk, were not being compensated. In order to find and solve the problem, Babcock developed a test in which milk was measured into the test tube. 17.6 milliliters of milk were usually taken in the test tube. Then 17.6 milliliters of sulfuric acid were added and centrifuged at 50 degrees. This resulted into floating of the fat at the top of the liquid. It could be measured. The principle behind the process is that everything in the milk except the fat dissolves in the sulfuric acid, and the centrifuge ensures complete separation with no bubbles in the fat. And the fat content can be measured using the graduation on the test tube. Conclusively enough, Stephen Moulton Babcock was American agricultural chemist and food scientist. He was born on the day 22nd October in the year 1843. Babcock spent much of his career at the University of Wisconsin and Agricultural Experimental Station. The dairy farming, cheese production, and cattle raising are the field of his contribution. He devised a very simple test for measuring the butterfat content of the milk. Release of the fat globule from the milk sample by dissolving the casein in the strong acid, and then separated the fat by means of centrifugation. The butter fat content could be read directly from the scale on the centrifugal tube. The Babcock test, as it was soon called, discouraged milk adulteration and provided for the first time an adequate standard for which fair payment for the milk could be determined. It also stimulated improvement of dairy production and aided in the factory manufacture of cheese and the butter. This is the list of few references. This presentation is dedicated to the memories of Stephen Babcock. Thank you very much.